Hi, welcome to Detours Understanding Acquired Brain Injury. And today we're going to talk a little bit about some of the more advanced MRI imaging techniques and how they are used in order to show some things that are not visible on the standard T1, T2 sequencing. Now, I'm going to talk about, and I've mentioned this before, diffusion tensor imaging and tractography. Now, the important thing to understand is this technique, it's been around for a little while now. It's been around for about 15 years, a little longer in the lab. However, the thing is, because insurance companies are kind of a little reluctant to pay for it, you generally do not receive this without special circumstances. So just go into your doc and say, hey, I want diffusion tensor imaging. I want tractography. I want you to, look at my white matter tracts or anything. Well, first of all, they're going to look at you kind of bug-eyed, most likely, if you see your regular PCP or talk to your regular neurologist about that. But because this is not something that is a common procedure for, the traumatic brain injury, it's usually done with research. There are some docs and there are some of the more advanced rehab hospitals that will do this, and it's maybe part of the research budget or some things like that. And maybe you can persuade a brain injury medicine specialist. These, as I've mentioned about these kind of docs before, they are neurologists, physical medicine docs, physiatrists, and psychiatrists who have a subspecialty in brain injury medicine. They are completed a fellowship. See my video on why my neurologist doesn't know about my brain injury. Unless they're one of these, a brain injury medicine physician, they typically don't know the nitty-gritty, the details. So, let's get into it. As I said before, an MRI is basically, it is different magnets that apply a field that generate an electromagnetic field and influence the way that hydrogen atoms are aligned, because hydrogen atoms are paramagnetic. They fall under, they have magnetic properties, and they align with or against a magnetic field. Not all of them, but a certain percentage. Obviously, you couldn't influence all of them like that. That would be one heck of a magnetic field. But it's enough that they fall under that magnetic field, a large enough percentage, and the ratio of water or other magnetic, paramagnetic, magnetic atoms in the body, that when they are energized using a radio pulse, they will receive the energy and then release it as given by the RF signal. Basically, the reason you use the radio wave is to energize it, and then you release it, and the hydrogen atoms then return based on conservation of energy, which is a principle that you have to give back energy that is given to a system. You can't keep it. And this is why, like, perpetual motion machines are impossible, things like that. You cannot create nor destroy energy. It's the same as with matter. Although there are certain loopholes, but there's always an accounting and a balance in our universe. Yeah, I know, nitpickers with philosophy and physics, there are certain loopholes and things, but still. So if you put energy into a system, it's got to return the energy. A certain percentage will come back to you as radio waves. A certain percentage will come back as heat. It's just the way it works, and we're not going into that right now. That would be ugly and very mathematical, and we're not doing that. But anyway, radio waves put energy in, and you get it back as a certain fraction of that energy. And the computers, there is always going to be a receiver. So transmitter puts energy in, puts RF energy in, and you have receivers placed inside the MRI machine. And you also typically, if you're doing one of the head, will have like a mask or whatever over your face, that grid-like thing or the different shape. That is a passive antenna. That is not transmitting anything. That is listening only. We discussed this in the MRI energy video. So now with DTI, what you're doing. Diffusion Tensor Flow. All right, let's get into what all this means. Diffusion of water. So what you're looking for is the behavior of water in the body. Now, the brain contains a lot of water, mainly in fatty tissues. Guess what? Well, first of all, the brain, 90% of it is not neurons. Only 10% of your brain is neurons. And there are differing ratios of white matter to gray matter. Now, gray matter is mainly just clusters of neurons packed tightly together. These areas, which are mainly on the cortex, on the outer layers, here, check out an image here of gray versus white matter. Here. Gray matter on the surface. White matter is the axons, which convey a signal. We've discussed this plenty of times, so I'm not going to repeat, but there are videos where I talk about diffuse axonal injury. The axons transmit. They're wires, excuse me, they're wires in effect in the brain. They convey the message. They convey an electrical current, electrochemical signal. It's not like electricity from your wall, but it's an electrochemical signal, which is conveyed down the axons. Axons are typically bundled together. And when there is damage, it affects how the axons, how they would appear magnetically. And that's what this is about. Diffusion tensor imaging relies on how water diffuses, how it travels along the axon. And that's what DTI is about. So it relies on what's called fractional and isotropy. So first of all, two different words. Fractional, you can figure that one out. But an isotropy refers to the fact that objects may have different, may appear differently or act differently depending on how you look at them, how you examine them, how you basically are like, for example, polarized light. Different perspectives as you pass it through a polarizer. Light has different properties depending on how you look at it. Wood grain. If you cut with a grain, it behaves one way. And if you cut against, it behaves another way. Okay? That's what it matters for an isotropy. So, and if something is, there are different ways. It either can be isotropic, which means it's the same in all directions. And when you're doing an MRI, a diffusion tensor flow, if water can move, for example, in all directions, you say it's isotropic. And you use a value of 0. So when you're talking about diffusion in the brain, what you're saying is that the value is 0. And the maximum value of free flow, you'll give it a value of 0. So water can go in any direction in the brain. Cerebrospinal fluid would be a good example of that in the ventricles, because its value 0. It can go in all directions, up, down, left, right, anywhere it wants to go. Brownian motion, which those of you who've taken, bio or, whatever, water can move any direction. Or if it is completely restricted, because, water can go any way it wants to, but within constraints of the restriction. Okay. If, on the other hand, it is channelized, it is forced to go in one direction only, its value is 1. And the more channelized, then you get a value of, like I said, 1. So this is a scale, and that is applied to diffusion tensor flow. Right, so it is a scalar number. It is a scale. Okay. Now, what you're doing with diffusion tensor flow, now that we talked about anisotropy, it's the measurement of like how restricted or how free can water flow in the brain. Now, why is this important? It's like kind of odd, a little esoteric to talk about it like this. The reason it's important is because water needs to flow through an axon, right? And food and, any kind of, because that's how they do it. And we talked about this, we talked about destruction of neurons. The cytoskeleton and the microtubules that take water and nutrients and other liquids down the body of the cell, down through the axon to the end, is channelized. It has to be. It's like a tube, like a drinking straw, right? So if there is damage to white matter, it's either completely restricted, a value of 0, or it's ripped apart and water flows any which way, value of 0. Okay, so if you don't get a value trending towards 1, if you get a 0 instead, that's telling you damaged axons. You're not getting free flow. Now, of course, you'd have to apply this to a map of the brain so you know it's not ventricles or outside, but anywhere else, you know you've got damaged axons. And axons bundle together to form white matter tracts. Another advantage is, now this is where tensor comes in. Using linear algebra. All right, remember, these computers are really good at math anyway, so you build what's called a tensor matrix. Tensor matrix is basically looking at these values of whether water flows directionally or whether it flows freely, whether it diffuses freely. You build a tensor, where T comes from. And so what you do is you build a matrix, which is like mapping the way water is flowing, left, right, forwards, backwards, and you build a map showing how water's flowing in the brain. Forward, backwards, left, right, and you then apply it to an image. And so what it shows is since you know X, Y, Z coordinates, those of you who remember from geography, or from geometry, not geography, although you could argue, geometry. Remember the Cartesian system, X and Y and Z, and then you apply that based on the ones and zeros, how far along that vector, that scalar system, each vector is, that point is, you know which way the tracts of white matter go. And we had talked about white matter tracts before. And so then you apply it to a map. You say, all right, these white matter tracts are going in this direction, these go in this direction, and these are all intact. And so I know that, the longitudinal fasciculus is intact, the corpus callosum is intact. You can then tell which specific tracts are intact. And so you can then complete a map, and then you overlay. And that is what diffusion tensor imaging is about. You then apply it to a graphics program and mapping deeper layers. And the colors, it's a very colorful, bright image showing you all the different tracts of white matter, and shows you whether there is flow or whether it is obstructed, whether it is blocked or whether it's free-flowing, whether it's a 0 or trends towards a 1. I mean, it's not perfect. There are obviously aberrations in everyone's brain because some neurons are damaged, some work, but overall, you could do a certain percentage and show, and different colors show away and towards for fluid flow, left, right. And so you can map like the corpus callosum, you can, like I said, uncinate fasciculus, you can map out projection fibers, callosal fibers left to right, and then intracerebral fibers. And then you can make some predictions based on which ones are intact, which ones are broken. You can look at healing, but you can also look at thickness of these white matter tracts, which tell you whether maybe someone has behavioral issues because perhaps certain fibers are too thin, and maybe they're going to have problems of processing speed. Maybe they're going to have affective problems because they can't make the association. If the uncinate fasciculus is too thin during developmental years, they may not be able to associate emotion with memories. And so they can't develop an affective attachment. They have problems with empathy. or applying affective memories backwards, or controlling their emotions because of thicknessing changes. They may have none at all. Again, this is why this is useful. It also shows after traumatic brain injury healing, whether new connections can be made for memory and things like that. And this is one of the reasons why diffusion TensorFlow is helpful. There are newer techniques that are available, Q vector spacing, QKI, and it's like there are other techniques which are more complicated but show movement within a certain space and allow you to see different types of tissue present within a given volume. And so you could tell, well, is this white matter, is this gray matter, is this, it's like to tell you density. And but these all have one thing in common, and they are still all anatomical scans. So it is important to keep in mind that as well. So here is another image of a DTI image showing damage. And since these are the anterior projections off the thalamus, this person will have emotional problems with, specifically with emotional processing and with executive dysfunction as well. And you can make predictions about specific mental health problems too. The thing is, when it comes to a lot of the newer techniques, while they may show interesting images and they may be applied to a lot of different software packages, in order to be useful, one of the things we've learned from MRI is it's like, if you can't apply it to some of the new discoveries, it kind of isn't very useful. It's like, it's great to have pretty pictures of somebody's brain and white matter. If you don't have proper mapping, great. It's kind of like a view from space or something like that of the Earth or moon or whatever. But if you don't have proper directional signs, things like that, it's just another pretty picture for some science book for like university neurobiology or whatever. Who cares? This is what tractography is about. And I want to show you tractography and connectome mapping. I do want to talk separately about connectomes, but tractography is the study of white matter tracts specifically, and how they interact with each other and how they build networks in the brain. Now, one of our tendencies when it comes to neuroscience and brain studies is this habit of looking at the latest tech thing and applying it to the brain and saying, oh, this analogy for the brain, whatever. But there are a lot of holes with that. Sure, they kind of make a decent analogy, but it's like, originally it was like, before then it was like the brain's a great big file system or whatever, back in the days and when it was all file clerks, and then it was like like a calculating machine, and then it was a computer, and now it's networks. But there is some correlation. We do know there are like networks of different gray matter nuclei that are linked by white matter tracts. And we do know if you disrupt, even if you do damage in one area of the brain that's not, like if you do damage in the back or parietal lobe, you can affect what seem to be frontal functions. So why a hit in the back might affect some frontal lobe, some behavioral or affective cognitive function. And you might say, well, gee, that's because contra coup injury, hit in the back, you do damage in the front. Yes, absolutely. But even some imaging shows that, wait a second, it's like they really haven't done damage in the front. It's like, but yes, if you disrupt a network, if you take out the temporoparietal junction, which is between the temporal and parietal lobe, you can affect somebody's ability to do theory of mind and to understand how other people think or how they may feel about your actions. And that's back further, back towards back of the head, and the ability to understand that people are more complicated than what you may believe. And so, and this relates to connectivity between that region and frontal lobes and things like that. So what is, a tractography? That is the study of, through the use of this kind of image and imaging to perform examination of how these white matter tracts connect. This here is is an example of that, Connectome tractography of mapping. And to develop a Connectome, I'm just going to touch on this, is they took thousands of people who have no psychiatric, mental health, those kind of problems, statistically looked at how their brains are wired at different ages, and got this map, this tractogram, got this Connectome of a healthy brain and how the connections are made between the different areas, between networks, between hemispheres, between deep brain areas, between cortical areas, between major centers responsible for performing different functions. Then you can compare other people who have different mental illnesses, who have different neurological disabilities, and then they looked at their behaviors, their thoughts, their cognitions, what they did, and over time. And so you stick someone in an MRI machine and you can say, oh, your pattern through matching, obviously because computers are aces at this, at saying, oh, this looks like this group of people after your brain injury, or while you're developing, you look like this. You look like you might have ADD, ADHD, or you look like you've got executive problems, or you look like you have a head injury, or you look like a narcissist, or you look like this or that. And you have 95% match with one of these. So obviously that is an ethical quandary, of course. What do you do with that kind of information? Do you deny a person like that a job in a certain career? Might they be harmful? They might not be aware of it. That's something else you could determine with Connectome map, performing tractography. They might not be aware they have certain deficits. They might deny it, either denial or absolutely unaware. They might not be aware of their own deficits, anosognosia, which you can also tell from this. And they might tell you they're fine, but you can look at this and say, nope, you're not fine. Or you have a tendency to be a pathological liar, however you wish to use that term. But to be a liar, you should be aware. There's no reason you shouldn't. All the connections are in place. You've got a personality disorder. Do we deny you certain jobs? Do we deny you the right to get married? You have a profile that looks like an abuser. Domestic violence. Do we deny you your rights and privileges? I don't know. Honestly. I don't know. But that's where we're headed. I don't know what that means, honestly. At least the ethical implications. I do know what, a map me like that means, but I don't know what it means ethically and where future leaders and where the youngsters want to take this. I do know that it can go a very bad place. If you've seen like Gattaca or many of these other interesting sci-fi movies, but I can tell you that things like fMRI, PET scans, other methods of scanning can uncover people who'd rather we not have them uncover them. We've always said genetics is not destiny. But are your brain connections destiny? Are areas that light up in your brain when shown images destiny? It's a quantitative EEG. Destiny? I don't know. What do you think? Because this would be a decision that affects all of us. Please comment below. Share, like, subscribe, express your opinion. Artificial intelligence applies to this kind of thing too. But artificial intelligence can't make our decisions like this for us. Or, at least if we don't make the right ones, maybe it will. Have a good day.